Hi, good morning. I'm Maria Maya Medalla from the Department of Anatomy and Neurobiology. I'm an assistant professor, and I want to tell you a little bit about the work we do in our lab. Our lab is interested in studying the cellular and synaptic dynamics of prefrontal circuits that are important for cognitive and emotional integration. So the key players for that network are the anterior cingulate cortex, shown here in the medial surface of the monkey brain, and the lateral prefrontal cortex, which are both involved in cognition but have distinct roles specifically with regards to their processing of emotional information, such that the ACC is more heavily interconnected with limbic structures that are involved in emotional processing, such as the amygdala and the hippocampus. So in our lab, we use cellular and synaptic techniques to study in high resolution the electrophysiology and the structure of pyramidal neurons in these two areas. So in a recent study I did with Jenny Lubke, we did whole cell patch clamp recording and immunohistochemistry to study the structure and connectivity, inhibitory and excitatory connectivity within these areas. And what we found is that the anterior cingulate cortex and the lateral prefrontal cortex differ markedly on their inhibitory circuits. Specifically, the ACC exhibited more diverse and a greater degree of inhibitory synaptic connectivity compared to the LPFC, such that the inhibitory neurons that are targeting these pyramidal neurons in the ACC showed longer synaptic inhibitory synaptic decay times, as well as a greater inhibitory conductance. And when we plugged in those cellular features in a model developed by Nancy Capel, this network model, we showed that these particular cellular features endow these two networks with distinct oscillatory dynamics, such that the ACC is more capable of having slower dynamic timescales in terms of their oscillatory behavior compared to the LPFC. Currently we're following up this work, understanding the role of the cholinergic neuromodulatory system in these two areas, as well as the role of distinct GABAergic cell types and receptors in these two areas. So a different focus of our project is also understanding how long-range extrinsic connectivity can affect this cognitive emotional integration, specifically within the ACC where we know they interact. And how we do this is first we can study the properties of the neurons that give out these pathways by combining the techniques that we talked about already, the cellular techniques, with neural tracing. So in this case, we can label projection neurons in the ACC directed to the prefrontal cortex versus the amygdala, and then we could study the intrinsic biophysical properties by recording from these neurons in vitro, as well as studying the structure of their dendrites. In addition, we could also understand the dynamics by which how information from the limbic system, such as the amygdala, can be delivered to the ACC and potentially passed on to the LPFC. In this sense, we combine these tracing techniques with optogenetics by injecting channel-rhodopsin in the amygdala, so we can study in vitro light-induced responses in cells that are labeled with tracer. So this is an example of one of our pilot studies where we recorded a pyramidal neuron in the ACC and injected sort of locally channel-rhodopsin in the same region, and then shown here in green, and we could see that the like responses in this specific neuron, excitatory versus inhibitory responses. And then the goal is to take these cells and connection circuits and look at them at the electron microscope to sort of understand the structure function dynamics of these networks.